Hello, everyone. This is Dr. Singh. Let us take a moment to understand what happens at the neuromuscular junction. The neuromuscular junction is a specialized synapse where a motor neuron communicates with a skeletal muscle fiber to initiate contraction. At the presynaptic side, the axon terminal contains vesicles filled with acetylcholine. Just below this lies the synaptic cleft, a very narrow extracellular gap. On the muscle side, the motor end plate forms a highly folded postsynaptic membrane, densely packed with nicotinic acetylcholine receptors. This precise anatomical arrangement allows rapid and reliable neurotransmission from nerve to muscle. It sets the stage for the electrical and chemical events that trigger a muscle action potential and ultimately muscle contraction. When the motor neuron's action potential arrives at the axon terminal, it depolarizes the presynaptic membrane. This depolarization opens voltage-gated calcium channels, allowing calcium ions, Ca+, to rush into the terminal, down their electrochemical gradient. Calcium acts as the trigger for exocytosis. Synaptic vesicles move toward the presynaptic membrane, fuse with it, and release acetylcholine, or ACH, into the synaptic cleft. Because the cleft is extremely narrow, acetylcholine diffuses very rapidly to the motor end plate below. In less than a millisecond, transmitter molecules reach and bind to nicotinic acetylcholine receptors. At this moment, the neuron-to-muscle chemical signal is complete, and the postsynaptic electrical response begins. At the motor end plate, acetylcholine binds to nicotinic receptors. These receptors are ligand-gated ion channels, and when acetylcholine binds, the channels open. Sodium ions rush into the muscle fiber, creating a local depolarization called the end plate potential. When this depolarization reaches threshold, it activates nearby voltage-gated sodium channels in the surrounding sarcolemma. This generates a full muscle action potential. The action potential then spreads rapidly along the sarcolemma in both directions and dives deep into the muscle fiber through the transverse or T-tubules. At this point, the chemical signal at the synapse has been converted back into an electrical signal inside the muscle. This ensures that excitation reaches deep into the fiber, where it can trigger calcium release and ultimately muscle contraction. The next step converts an electrical signal into a chemical trigger inside the muscle fiber. When the action potential travels down the T-tubule, it changes the voltage across the T-tubule membrane. Dihydropyridine receptors, also called DHP receptors, sense this depolarization. In skeletal muscle, these DHP receptors are physically coupled to ryanodine receptors, or RYR channels, on the adjacent sarcoplasmic reticulum. When the DHP receptors are activated, they pull open the ryanodine receptor channels. This allows calcium ions, Ca2+, to flood out of the sarcoplasmic reticulum and into the sarcoplasm. The result is a rapid rise in intracellular calcium, increasing by nearly 100-fold. This surge of calcium primes the muscle's contractile machinery. Without this calcium release, myosin cannot effectively interact with actin, and muscle contraction cannot occur. Here we zoom in to the molecular engine of muscle contraction. Calcium binds to troponin on the thin filament. This binding shifts tropomyosin away from the actin filament, uncovering the myosin binding sites. Energized myosin heads then attach to actin, forming structures called cross bridges. Next comes the power stroke. The myosin head pivots, pulling the actin filament toward the center of the sarcomere. When ATP binds to myosin, the myosin head releases from actin. ATP is then hydrolyzed, which recocks the myosin head, positioning it for another pull. As long as calcium levels in the cytosol remain elevated and ATP is available, many cross bridges cycle continuously. These cross bridges do not move all at once, but asynchronously, producing smooth and sustained tension and muscle shortening. This repeating ATP-driven cycle is the mechanical payoff of the earlier electrical and calcium signals. Hundreds of myosin heads on each thick filament work in parallel. Because of this, microscopic filament sliding scales up to generate force across the entire muscle fiber. Contraction stops when the neural signal and intracellular calcium are removed. First, acetylcholinesterase rapidly breaks down acetylcholine in the synaptic cleft. As a result, the end plate potential ends and muscle action potentials stop. Without new action potentials, the DHP-ryanodine receptor pathway closes, and no additional calcium is released from the sarcoplasmic reticulum. At the same time, SERCA pumps in the sarcoplasmic reticulum membrane inform, actively move calcium ions back into the SR. This lowers the level of calcium in the cytosol. As calcium levels fall, calcium detaches from troponin. Tropomyosin then slides back over the myosin binding sites on actin. Without exposed binding sites, cross bridges cannot form. Force production declines, and the muscle fiber relaxes. This process highlights an important dependency chain. Neuron action potential leads to acetylcholine release. Acetylcholine produces an end plate potential. The end plate potential triggers a muscle action potential. The muscle action potential causes calcium release. Calcium enables cross-bridge cycling, and cross-bridge cycling produces contraction.